Hello viewers, today we are going to discuss about a topic that is hybridoma technology that involves production of monoclonal antibodies by using hybridoma cells is commonly called as hybridoma technology. This hybridoma technology was introduced for the first time by Kohler and Milstein in the year 1984. They got Nobel Prize for their work, and this technology is mainly useful for production of monoclonal antibodies. So the principle of this hybridoma technology is hybridoma cells are prepared. With the help of these hybridoma cells, monoclonal antibodies which are having applications in various fields of biology will be produced. So what is hybridoma? Hybridoma is a hybrid cell formed with a combination of lymphocytes and myeloma cells. The hybrid cell formed with a combination of lymphocyte and myeloma cell is commonly called as hybridoma, and production of monoclonal antibodies by using this hybridoma cell is called hybridoma technology. So you can see in the slide the production of monoclonal antibodies. So here hybridoma cells are prepared first, and with the help of hybridoma cells the monoclonal antibodies are generated. So production of monoclonal antibodies by using hybridoma cells consists of two main events. One is preparation of hybridoma cells, second one is production of monoclonal antibodies. So preparation of hybridoma cells consists of 6 steps. First one is immunization of specific animal which generate hybridoma cells. Second one is isolation of myeloma cells. Third one is fusion of spleen cells with myeloma cell, and fourth one is selection. Fifth one is isolation and screening of hybridoma cells. So otherwise the steps can be shortened like isolation of B lymphocytes, isolation of myeloma cells, fusion of B lymphocytes with myeloma cells, selection of hybridoma cells, screening of high productive hybridoma cells. So these are the various steps in preparation of hybridoma cells. So first one is isolation of B lymphocyte. It is a very simple step. Here a animal, in vitro grown animal is selected. Into that animal known antigen is injected. The antigen for which we need monoclonal antibodies are injected into the in vitro grown animals like mouse. So after injecting the antigen, the mouse is killed and the spleen is taken out, and the cells of the spleen called splenocytes are isolated, and these splenocytes are nothing but B lymphocytes, antibody producing B lymphocytes. And second step is isolation of myeloma cell. Here, so myeloma cells are fast growing cells isolated from bone marrow, and these cells will divide rapidly. So these myeloma cells are isolated. So in the first step, B lymphocytes capable of producing antibodies are isolated. In the second step, myeloma cells are isolated. Coming to third step, so B lymphocytes and myeloma cells are fused. Before fusion, the B lymphocytes and myeloma cells, the myeloma cells are treated with 8-azoguanine, and these 8-azoguanine will make them to remove HGPRT from the myeloma cells. So B lymphocytes are fused with HGPRT minus myeloma cells. That is commonly called as somatic cell fusion. So once the fusion is over, the selection of hybridoma cells. See, when we take B lymphocytes and myeloma cells and when we fuse, we get different types of cells. So a B lymphocyte may fuse with B lymphocyte, a myeloma cell may fuse with myeloma cell, a B lymphocyte may fuse with myeloma cell to form hybridoma cell, and unfused B lymphocytes, unfused myeloma cells. So we get 5 combination of cells. Out of these 5 combination of cells, we need to select a cell that is formed with a combination of B lymphocyte and myeloma cell. That cell is called hybridoma cell. So how to select these hybridoma cells from other cells? That can be done with the help of 2 pathways. One is salvage pathway, second one is de novo synthesis. By using any one of these, we can able to select hybridoma cells from other cells. So once the hybridoma cells are selected, generally hybridoma cells are selected by using hat medium. So in hat medium, only cells containing HGPRT will grow. So B lymphocytes and a hybridoma cell, myeloma cell combination will grow on hat medium, will grow fastly. So the cells which grow fastly in hat medium are selected and used. So once the, you can see, so myeloma as they are not having HGPRT, they cannot grow on hat medium, and B lymphocyte they grow, but they grow slowly. Unfused B lymphocytes, unfused myeloma cells, unfused B lymphocytes will grow but slowly, unfused myeloma cells cannot grow. So the cells which grow and which grow fastly are hybridoma cells, and these cells are selected and subjected to screening. So the screening involves usage of ELISA. So here what we do is, we take a multi-well plate. The multi-well plate will be taken, and the multi-well plate is coated with the antigen that we introduced into the in vitro grown mouse in the beginning, and selected hybridoma cells are introduced into this multi-well plate. Okay. So once we introduce the hybridoma cells into multi-well plate, then antibody producing hybridoma cells will be binding to the antigens, and after washing, unbound hybridoma cells will be washed off. So then we have to add an enzyme, an antibody linked to enzyme. Okay. So this antibody will go and bind to antibody, and antibody is linked to antigen. So then we have to add an enzyme. So then we have to add the substrate for the enzyme. Then the substrate is added. The substrate will be converted into colored form by the activity of enzymes. So the wells which show the dark color indicates high productive clones, and these clones are selected and used. So by this way we can able to produce high productive hybridoma cells. So this is the part 1 of hybridoma technology. So in the coming classes we will discuss about production of maps by using hybridoma technology.